This conference will now be recorded.
Okay. So I hope many of you have arrived. We'll see another video in which we can target drugs because basically today's class is on lab diagnosis and treatment. So this will help you in understanding what targets are used for treatment options. Okay, so you already know about HIV replication, what are the different targets, and all these things. So today we'll discuss whatever has been discussed before. We'll have a quick recap, and then we'll move on to lab diagnosis and treatment in very short. Okay. So I hope I'm audible. Anyone can just unmute and say, is the screen visible and am I audible?
Yes, ma'am.
Yes, ma'am.
Okay, thank you. Thank you so much. Okay, so today's class is HIV 2, the basic things which we have learned before. We'll have a quick recap of that, and then we'll move on to the lab diagnosis. So basically, there has been an emergence in pandemic spread of acquired immunodeficiency syndrome, and this has been the greatest challenge to public health until COVID-19 came, that is. And how it was known that in the late 1970s and early 1980s, an unusual number of homosexual men, Haitians, heroin addicts, and hemophiliacs. So the 4H, you can see there are 4H. So these 4H club in the US were noted to be dying of normally benign opportunistic infections like pneumocystis carinii. That time, this was only a very minor type of infection, but they seem to be dying out of that. And there were similar opportunistic infections which were not that much dangerous as such in normal people, but in these particular group of people, that is these homosexual men, atheists, and heroin addicts and hemophiliacs, they started dying out of those so Luke Montagnier and colleagues from Pasteur Institute in Paris in 1983, they isolated a retrovirus. Why is it called a retrovirus? You have seen that there is something known as a different type of known as retro means going back. Yes, so from DNA normally it proceeds to RNA and then into proteins. That is the central dogma which we already know. But here what is happening is from RNA it is changing into DNA, and then that DNA is getting incorporated into the host cell DNA. So there is a big change from the normal thing that happens. So that is why it is known as a retrovirus. And this was isolated from a West African patient with lymphadenopathy. So Robert Gallo and colleagues, they isolated this retrovirus from AIDS patient and called it HTLV3. And later on, they knew that these two viruses are the same. So later on, the generic name was given by the International Committee on Virus Nomenclature, that is HIV, Human Immunodeficiency Virus. So it has been seen that it has evolved from a simian virus, from a monkey virus, onto humans and spread through Africa and the world by an increasing mobile population, because now the population is quite mobile. So it's very easy for any infection to spread very easily. So basically it is an enveloped spherical virion, as we saw in the video. It is about 80 to 120 nanometers in diameter, and there are 2 copies of single-stranded positive sense RNA. And there is a very typical enzyme. Please remember the name of this enzyme. This is an RNA-dependent DNA polymerase or a reverse transcriptase. So it goes reverse. So from RNA, it changes into DNA. So it changes the RNA of this particular virus into DNA. So that is why it is known as an RNA-dependent DNA polymerase. This particular enzyme is the reverse transcriptase. And there is a virus receptor, which is the initial determinant of tissue trauma. DNA and there is a replication through an intermediate and video. So that particular DNA intermediate is known as the provirus, and this integrates randomly this particular for many years. You might have seen some patients initially they might be infected, maybe HIV. Sometimes they may have maybe something like a flu, but later on it will change into of HIV, and then it take from 10 to 30 years for this progress to happen, and that is because this provirus will integrate randomly. It will, from time to time, it will help in the replication of the virus, and that is why this thing happens. So there are important viruses in this retroviridity family, like HTLV1 and 2 and HIV. And we can see here that lentiviruses. These viruses are known as lentiviruses. So there are class 1 and class 2 different viruses, class 3 viruses of retroviruses. So this particular HIV belongs to lentiviruses. Here you can see HIV1 and HIV2. So lentiviruses basically causes, they can cause slow virus disease. Slow viruses also will be taught to you. So like Bisnar maybe in sheep and equine infectious anemia, and immunodeficiency causing viruses like HIV1, 2, and simian immunodeficiency virus causing simian AIDS, and feline T lymphotrophic virus causing FAIDS. So these are the lentiviruses. Here you can see these are the different groups of lentiviruses and other viruses also. So different types of retroviruses. So lentivirus is a subgroup of the family Retroviridae, and these are spherical in envelope virus, about 90 to 120 nanometer in size, and the nucleocapsid has an outer icocycloid shell and an inner cone-shaped core, which we saw in the video, and the genome is diploid and it has two identical single-stranded positive sense RNA copies, and in association with RNA, the reverse transcriptase enzyme, which is typical to this virus. So there are important parts. Everybody has to remember the diagram of HIV and be able to draw the diagram of HIV, because this is quite important. So here you can see the two strands of a single-stranded positive sense RNA, and in association with that, you can see that reverse transcriptase enzyme is present. There are other enzymes like protease, which we saw in the picture. It cleaves the proteins which are formed, and then that cleavage is also very important, and that can be also a target for drugs. And at the same time, you can see that matrix protein P17 or P18 is present. So that is also important, because we can help in knowing about the virus. And these are the different peplomers which are present, that is GP120 and GP41, and this is like a pedestal which is present on the virus. And then there is the capsid protein P24, which helps in early diagnosis. So in the window period when antibodies have not formed, in that stage you can diagnose only with the help of P24. So there is a window period in which it takes time for the antibodies to appear, and in that time we can detect only the antigens, and that antigen among them, one is P24. This is a very important antigen. So P24 antigen is an important antigen. This is the capsid protein. So that these parts are important. So everybody has to be able to draw this diagram and know which parts are important. And then there is the enzyme integrase. Here you can see this helps in integrating the proviral DNA, that is from RNA, whatever is produced, the DNA that is integrated into the host cell chromosome. So that enzyme is the integrase. So these are the different parts of the HIV virus. So these are different genes which code for structural proteins, and these genes are helpful in diagnosis through PCR, or suppose quantitative PCR you want to do, or even qualitative PCR. So suppose you want to detect the genes, then you can help in knowing that this is the GAG gene, the ENB gene, and the POL gene. So these are the three genes which are important. There are other genes which are also given in your textbook, but we'll concentrate only on these three. So GAG gene is the core and the shell of the virus is formed through this gene, and the major antigen, major core antigen which appears, that is P24, which can be detected in the early stages before the antibodies appear. So that is important. So ENV is the envelope protein. So this is the surface spike, that is the GP120, and the GP41, that is the transmembrane ankyrin protein. And then there is the POL gene, that is the polymerase or the protease and the integrase enzymes. So these enzymes are formed through the POL gene. So these three genes you have to remember. One is the GAG, one is the ENV, one is the POL gene. So here you can see this is the gene. So the GAG, POL, and ENV. So other repeating, not repeating parts of the chromosome is also there, but we concentrate only on these three. So one basic thing we have to know about our own immune system in order to understand why there is immunodeficiency. So what happens basically, this is a general thing which we are discussing. So to just recap what you learned already in your immunology classes. Whenever any antigen, that is a foreign material, enters into the body, it is basically, it cannot be detected by the T helper cell. The T helper cell, the T cells, they need the help of other cells, that is the antigen presenting cells. So they will present the antigen after cleaving it. So here you can see this antigen is cleaved, it is cut into different pieces, and then it is basically presented. So somebody has to tell the T cells that this particular antigen has arrived. So once this antigen presenting cell, which can be a macrophage, which can be any other cell, so which can present, this is an antigen presenting cell, so through MHC2 it will present. So I hope you remember about major histocompatibility complex. So through MHC2 it will present to the T helper cell, that is the CD4 cell, and the CD4 cell, then only it will come to know that any particular antigen has arrived, that is any foreign material has arrived. And once it knows that it has arrived, then the T helper cell plays a major role in our immunity. How it will go and it will tell other cells like the B cells to produce the antibodies by becoming plasma cells, and then it will go and tell other macrophages also, and it will go and tell other killer T cells also, it will go and tell cytotoxic T cells also, it will help in the memory T cells, the memory B cells in building up also. So lots of cytokines, other things are also released by the T helper cell, and thus the T helper cell is basically a helper cell, that is it helps other cells from knowing that this thing has arrived. So basically when the antigen arrives, the T cell doesn't know. So it has to be presented by an antigen presenting cell, and once it has been presented and once the T helper cell has recognized that a particular foreign material has arrived or an antigen has arrived, it will go and tell about that to all other cells. So that is why it plays a key role in our immunity. And imagine if this particular thing is not there. Imagine if the T helper cell is not there, or if it is less in numbers. So what will happen? So naturally, whatever benign or whatever normal infections which should subside in maybe 2 days or maybe a week, it will go on increasing, it will go on increasing till the patient has died from opportunistic infections. So basically, whenever a patient has AIDS, that particular patient does not expire or does not have a problem with HIV, but basically with opportunistic infections. And why do those opportunistic infections happen? Because these T helper cells are no longer there, because they are targeted by the HIV, and that is why this thing escalates into a new situation. So basically, whenever a virus enters into the blood or tissues of a person, it comes in contact with a suitable host cell, and the CD4 antigen is the receptor. So the CD4 T lymphocytes like the T helper cell or the inducer cell, and up to 5 to 10% of B lymphocytes, 10 to 20% of monocytes and macrophages, and glial cells and microglia in the CNS. So these particular cells, all of them can be the target of the HIV, but the important target is the CD4 T lymphocyte. So that is why it is put in red. So CD4 T lymphocyte is a very important target of the HIV. So here you can see there is a receptor and a co-receptor. So GP120 and GP41, both of them, they go and they will bind to the receptor, that is the CD4 T lymphocyte, and CD4 is basically, it is there on the cell, so it will go and it will bind to the CD4 receptor. So there is specific binding of the virus to the CD4 receptor by the GP120, the glycoprotein 120, and there is participation of the co-receptors like CXCR4 in T cell tropic strains and CCR5 in macrophage tropic strains, and then there is fusion of the virus with entry cell with the cell and entry into the cell, and then there is uncoating of the genome. So here you can see this is the receptor, and these are the co-receptors. So here you can see GP120 and GP41. So the receptors and the co-receptors, both of them, they bind, and after binding it can only help in, after co-receptor binding it will help in the virus cell fusion. So whenever the virus infects the cell, the viral RNA is transcribed by an enzyme that into the single-stranded DNA and then into the double-stranded DNA. That is the reverse transcriptase enzyme. So once the reverse transcriptase enzyme produces the double-stranded DNA, that is the provirus, this provirus is integrated into the host cell chromosome with the help of the enzyme integrase. And then the provirus remains latent for long periods, and then this can influence host cell function. So here you can see through this is the HIV RNA, and through reverse transcriptase it is changed into HIV DNA, and this particular HIV DNA will go and get integrated into the host cell chromosome with the help of the enzyme integrase. So here you can see the virus particle has arrived. This is the capsid. So here this is changing into the DNA, and once the RNA is changed into DNA, that will go and get integrated into the integrated proviral DNA. So, and because this is integrated from time to time, it will go on producing the HIV particles, and that is why for a long time this HIV infection will remain in the patient, and it is very difficult to eradicate. So, in response to viral promoters, the provirus initiates viral replication by directing the synthesis of viral RNA and other components. And once the virus buds out of the lipoprotein envelope, the envelope consists of the lipid derived from the host cell membrane and virus-coded glycoprotein. So this happens not only in HIV but other viruses also which contain the lipid envelope. So lipid is basically derived from the host cell membrane. It takes basically a pinch from the host cell membrane and it will take, as you saw in the video. So all these virus-coded glycoproteins will be inserted into the particular part which is to be the envelope, and then it will take a pinch and then it will. So there is a long and variable incubation period of HIV infection, and this is due to latency, and this latency is because of the integrated proviral DNA. So what happens is HIV can be isolated from blood, lymphocytes, cell-free plasma, semen, cervical secretion, saliva, tears, urine, breast milk, etc. So it will go on get producing for years together. And because there is major damage to the cellular immunity, and because of lack of proper T helper cell activity, there is a polyclonal activation of the B cells, and there is hypergammaglobulinemia. There are a lot of immunoglobulins which are produced, but these immunoglobulins are useless. Why they are useless? DNA the provider has remained inside the cell and they can do nothing. So a lot of useless immunoglobulins are immunoglobulins. So monocyte macrophage function is affected, there is intracellular killing is diminished, and there is failure of immune response because the helper T cell activity is affected. So this you have to remember. So HIV is a highly mutable virus, and because of frequent antigenic variation, there are differences in the nucleotide sequences, the cell tropism, the growth characters, and the cytopathology. And the original from HIV-1 find in our hospital also. So there is less genetic identity of HIV-2 with HIV-1, and HIV-2 was basically found in West Africa, and it is closer to the simian immunodeficiency virus. So you can see the adult HIV prevalence rate is quite high and it has increased over the years. And at least 10 subtypes are there based on the sequence analysis of the GAG and the ADNV genes, and there are subtypes designated from A to J, and major different groups are there, that is M, O, and N, outlier and mean. So majority of the worldwide are caused by the subtype M, and O is basically found in West Africa, in Cameroon and Gabon, and N is found in Cameroon and distinct from the other two, M and. So the importance of antigenic differences is that there is immune escape. Suppose something goes on changing, we don't exactly know that what is the particular antigen that is there, and because of immune escape, there is the major problem. So serodiagnosis is basically done, and because of subtypes in Asia and Africa, because of heterosexual contact, and American strains are also different. So depending on that, we have to have a very global type of diagnosis. We have to have different types of methods in which we can diagnose all the different types and subtypes. So there are different stages, like the primary stage in which there is a short flu-like illness. So HIV, basically they will suffer like a cold or something. They won't even know that they are having HIV. And in the next stage, also they will be asymptomatic, and it can last for an average of up to 10 years. And the patient will be free from symptoms. There may be neuropathy, and HIV antibodies may be detectable in the blood. And then there is a symptomatic stage in which the symptoms are mild and slowly the immune system deteriorates, and slowly there is an emergence of opportunistic infections and cancers. And then there is the last stage, that is AIDS, in which the immune system weakens and the illnesses become more severe, leading to an AIDS attack. So opportunistic infections, a very long list, very long list is there, out of which only the important ones are bacterial infections, like examples. These are only examples, like tuberculosis and pneumonia, and then Kaposi sarcoma, herpes zoster, influenza, and then there is pneumocystis carinii infection, Candida cryptococcus. So they can be spread through many modes, as you know, through injectable routes, through blood, from mother to baby. So different routes are there. So through bodily fluids, basically different body fluids can transmit the infection, and through IV drug use they can be transmitted by sharing needles, drug users. So they are also a major part of the HIV infected population, the people that be living with HIV, and increase the chance of contracting HIV. So from mother to baby also it can be transmitted before birth, during birth, and post birth. So now we'll go to a little bit of testing options for HIV. So basically testing is anonymous. Please remember this, that no name is used. The patient remains anonymous. There is a unique identifying number, and the test results are issued only to the test recipient. And then there is a counseling session. There is a pre-test counseling, and there is a post-test counseling. It is all confidential. The results are issued only to the test recipient. And in pre-test counseling, the patient is told all about the transmission, prevention, risk factors, voluntary and this is voluntary. Remember that you can't force somebody to undergo the test, and it is always confidential. You can't go and tell their spouse. So all those things are highly confidential. So reportability of the positive test results is also very much important. So post-test counseling also, the clarifying of the test results, the need for additional testing, whether additional testing is required or not, whether CD4 cell count is required or not, and safe behavior. We have to tell them that this type of behavior you have to do, this you have to do, this you don't do. So all those things you have to do, and then you release the results. So there are different tests for detection, out of which only the important methods we discussed, that is ELISA, enzyme-linked immunosorbent assay, that is basically done for the antibodies. And then there is RIA also, so different radio immunoprecipitation assay is also there. Polymerase chain reaction is also there, and Western blot confirmation test is also there. Out of which I'll just remind you, you might have read already about ELISA. Just I'll put a quick recap. So whatever we want to detect, that is the antibody. Here you can see that is in green. So that is there in the patient's serum. So what we do is we coat the wells. We coat the wells with the particular antigen against that particular antibody. And once we coat it, then what will happen is, once we put the patient's serum, naturally the patient's antibodies will come and bind to that particular antigen. And once it binds, then what we'll do is we put a secondary antibody which is linked, so it is enzyme linked, so it will be linked to a particular enzyme. So this is linked, this particular secondary antibody is linked to an enzyme, and then what we do is we put the substrate to that particular enzyme, and once it fluoresces or some sort of color change is there, so once the color change is there, we can detect easily. So here you can see the antigen is coated on the wells, and then you add the specific antibody to be measured, that is through the patient serum. This is there in the patient serum. So once it is there in the patient serum, naturally it will bind, and every step is having a wash. Why is it having a wash? Suppose the antibodies are not there, then they won't bind. So what will happen is they will not bind to the antigens. So if the patient's antibodies are there, somebody has not muted their mic. Please take care that you mute yourselves so that the thing is always clear. So then the specific antibody is added through the patient serum, and then it is washed, and then the enzyme linked antibody, the secondary antibody is added, and then we put the substrate after the wash. So after putting the substrate, there will be a color change, and how much amount of color change has occurred, you can measure through a particular wavelength of light, that is in some particular nanometer of wavelength of light you put, and then you measure how much amount of antibody is present. So it is qualitative as well as quantitative. So this is about ELISA. Then there is Western blot. Western blot is basically, you might have heard about Southern blot, Northern blot, and Western blot. So Southern was actually taken from a name of a person, so that is for DNA, but Western blot is for proteins. So here what we do is basically in Western blot, what we do is we do electrolysis. So once we do electrolysis, what will happen is according to their weights, basically according to their molecular weights, components will be separated. The antigens or particular proteins will be separated. So once they are separated, here you can see that at different, there is the cathode and the anode. So in electrolysis, what will happen is the more amount of the lighter the molecule, it will travel more. That is basically in layman's terms how it will go. So once what will happen is it will go to more distance, it will travel, and then you can find bands. So here you can see that what has happened is the proteins have separated. So the proteins have separated here, and once they have separated, why there is the term blot? Because we have a nitrocellulose paper and then we blot it. Okay. So once we do the electrolysis and all the components have separated, we blot it onto a nitrocellulose membrane, and then once we have separated on the nitrocellulose membrane also, we are having the separated proteins. And once we have the separated proteins, what we can do is the same principle as ELISA. We can put a primary antibody, and then we can put a secondary enzyme conjugated antibody, and then we can put an enzyme and a substrate, and then we can detect through a detection signal. It can be colorimetric or it can be luminescent. So this is the detection in Western blot. We also have a short video on that so that we can understand. So Western blot is also important. Please remember, suppose a question comes on HIV, do not forget to write all about the pathogenesis. You write about different stages, you write about the lab diagnosis, you write about ELISA, you draw diagrams, you write about Western blot, draw diagrams. So all these are there. Okay. So why is diagnosing important? Because we have to start early and effective therapy, and we have to prevent the spread of disease. So early diagnosis naturally will allow for early treatment, and more evidence will show that this will improve disease outcome, and effective treatment also decreases the likelihood of spread, and the partners of the individual can also be tested. So that is why targeted testing is done for recent HIV exposure, symptoms which are suggestive of new HIV infection. Suppose there is occupational exposure, suppose there is a nurse who is having a needle stick injury, so for them we have to test. So blood or organ donors, and in pregnancy every time we have to test for HIV. So how is the diagnosis made? So basing on the signs and symptoms, clinical history, and it should only prompt the suspicion of infection, and it is confirmed with laboratory testing. So we have to take the proper history of exposure. We have to take the symptoms like constitutional and other symptoms like lymphadenopathy, other symptoms which can be present, and many may not also experience symptoms. So in the window period, we have to detect for P24 antigen. So window period means the time before the antibodies appear. Why there is a window period? Why it is known as a window period? Because there is a time difference in which the antibodies have not yet appeared. Because for the antibodies to form, as we saw in the picture in which the T helper cells are going and telling the B cells, you please produce the antibodies, all those things to happen, it will take time. So that is why there will be a window period. So in that window period, we can detect the particular virus with the help of P24 antigen. So that is why, suppose you are having a blood donor in a blood bank, suppose you are suppose you are taking blood for donation. Suppose that patient is testing negative through ELISA for antibodies, even then you can test for P24 antigens. So, what are the treatment options? In very short, there are different types of antiretroviral drugs. As we saw in the video, different targets are there. So, targeted nucleoside reverse transcriptase inhibitors and non-nucleoside reverse transcriptase inhibitors are there. So, zidovudine and protease inhibitors are also there. Basically, what we are inhibiting is the reverse transcriptase and then the protease, the enzymes, as you saw in the video. So anybody who is interested can separately ping me, or through the CR they can ping me. I can share the slides with you. I can share the the videos with you. Those videos are readily available on YouTube. If you want to see also, you can readily click and then you can see the videos. So basically the treatment is you have to treat the opportunistic infections also. So what I want you to do is I want you to write an essay. It is not a long question. It is an essay on HIV. You have to think about what the patient is suffering. You have to think about the patient's attendance. You have to think about counseling. So everything you have to think about, and then only you have to write an essay on HIV. So you can send the essay on my WhatsApp number in a Word document or whatever document you think fit. It can be a PDF document also. So please send it to my WhatsApp number. This is my WhatsApp number, or to my email ID. It's my email ID, and then we'll see a short video on this particular Western blot. So with that, we'll finish. You understood? I hope the basic things about HIV. If we start speaking about HIV, it will take some few days. Okay, just see how Western blot is done. Okay. So I hope you understood basically about HIV, about Western blot, about the different steps in which HIV will replicate. So please write the essay on HIV. It can be a sort of reflective essay if you have had any experiences, if you have gone to the wards and seen any patient, or suppose you have seen any movie, whatever you have thought about. So add your thoughts. The more reflective, the more amount of thinking that you do on your own, it will be a better essay. Don't just make it like a long question answer. So I hope that you write it and you'll send it to me. So I hope that all of you are safe in your homes along with your parents and your siblings, and take care of yourselves and don't go out. So thank you for joining us today, and have a good day.